Hello students, I am Dr. Sapna Kumari, Department of Zoology, Patna Women's College. Today we are going to deal with skeletal muscle contraction. We all know that muscle is made up of skeletal muscle tissues, connective tissues, nervous tissue and blood. As far as features of skeletal muscles are concerned, it is long and cylindrical in shape. It is multinucleated, which means it has many nuclei per cell. Striations are prominently visible because of the presence of alternate light and dark bands. Skeletal muscles is voluntary in nature, which means it is under our control. It is responsible for rapid contractions. They are attached by tendons to the bones or occasionally to the skin. Under the electron microscope, skeletal muscle is visible as shown on the slide. This is the ultrastructure of a skeletal muscle. Each muscle fiber is a single, long, cylindrical muscle cell. Sarcolemma is the cell membrane of the muscle cell, which has invaginations that penetrate through the cell called transverse tubule or T-tubule. Sarcoplasm is the cytoplasm of the cell with many mitochondria and nuclei. It has myofibrils. Myofibrils are separated into compartments called sarcomere that contain thick filament and thin filament. Thick filaments of myofibrils are made up of the protein myosin, while the thin filaments are made up of the protein actin, which is bean-shaped. The organization of these filaments produces striations. Furthermore, a sarcomere extends from Z line to Z line. I bands or isotropic bands are light bands made up of actin and anchored to Z lines. A bands or anisotropic bands are dark bands made up of overlapping thick and thin filaments. In the center of A band is H zone, consisting of myosin filaments only. In the middle of H zone, there is M line, which functions to hold the thick filaments to one another. Widely accepted, the sliding filament model for the muscle contraction was proposed in the year 1954 by the scientists H.E. Huxley and Hansen and A.F. Huxley and Niedergerg independently. Myosin and actin filaments lie side by side in the A bands, and in the absence of ATP, they do not form cross linkage. During contraction, actin filament slides over and move into the A bands and the H zone is filled up. The I band shortens, the Z lines comes in contact with A band, while during stretching, only the I bands and H zone increases in length, while A bands remain the same. Myosin filaments remain in the relatively constant length during the muscle stretch or contraction. Now, let us see the molecular mechanism behind the muscle contraction. As nerve impulses enter the presynaptic terminal of the neuromuscular junctions, the impulse causes acetylcholine to be released from the synaptic vesicles in the axon terminal. Then, acetylcholine diffuses across the synaptic cleft and opens sodium ion channels in the muscle membrane. Na+ enters the muscle cell and depolarizes it. T-tubules carry impulses into the sarcoplasmic reticulum and releases calcium ion. As calcium ion enter the individual muscle fibrils, it binds to troponin molecules on tropomyosin strands. This brings about the conformational change in the tropomyosin strand and exposes myosin binding sites on the actin filaments. Myosin along with ATP then binds to the actin, forming cross bridges. ATP breaks down, releasing energy, causing cross bridges to pull actin strands. Another ATP binds to the myosin cross bridge for the recovery stroke. Bend, attach and pull on the actin strand. When the action potential ends, calcium ion pump back into the sarcoplasmic reticulum. Tropomyosins covers the binding sites and myosin can no longer bind. Here on the slide, you can see in the figure A that how tropomyosin covers the myosin binding sites on the actin filament and makes inaccessible for myosin head in the absence of a stimulus during muscle relaxation. While in the figure B, you can see that in the presence of a stimulus, calcium ion binds to troponin complex and brings about conformational change in the tropomyosin, which in turn uncovers the myosin binding sites on the actin filament. In this figure, myosin head along with ADP and inorganic phosphate in high energy configuration first attaches to actin filament and forms cross bridge. Then in power stroke, cross bridge pulls the actin filament sliding towards the M line and releases ADP and inorganic phosphate. Now, new ATP attaches to the myosin head and causes the cross bridge to detach in low configuration. Then, in next step, ATP splits into ADP and inorganic phosphate, which provides power to cock the myosin cross bridge. In this way, contraction cycle goes on repeating until the stimulus is there. There was contraction in one sarcomere. Similarly, contraction in all the sarcomere throughout the length of muscle fiber brings about overall contraction. It should be also noted that for completion of one contraction cycle, 2 ATP molecules are required. Now, I hope this topic must be clear to you all. Thank you.